talk to you today with a focus on Ebola virus. Although, if you don't want to talk about Ebola virus and you have other questions, feel free to chime in on different topics. I think there have been a lot of questions about Ebola virus, and primarily I think it's because this is not a virus that's in the United States, or it has not been in the United States. This is a virus that originated from Africa. And to be perfectly honest, we actually still don't know a lot about this virus. Part of the reason is that because most of the outbreaks have actually occurred in Central Africa, in fact all of them, nearly all of them have occurred in Central Africa, they've typically occurred in remote villages, where usually usually you identify a few cases, a small cluster of cases, and then quickly there are efforts by non-governmental organizations, the World Health Organization, the CDC, to go and intervene, stop the outbreak. And so it's been contained largely for the roughly 30 or so outbreaks since Ebola virus was first discovered in 1976. The problem right now, though, is that this is an outbreak that basically went out of control. And we'll sort of talk about that and sort of what implications does it have for us here regarding Ebola virus. So let's talk about the origin. Like, where does this virus come from? We actually don't even know the natural reservoir of this virus. It is suspected that it's fruit bats, and there are a couple of reasons why. If you actually look at fruit bats, you'll find that they have specific antibodies to Ebola virus. You can find Ebola virus sequences in fruit bats. No one has ever been able, though, to actually identify infectious virus in a fruit bat. However, they have been able to identify a relative of Ebola called Marburg virus. These are viruses that belong in a category called filoviruses. And Marburg viruses, or live Marburg viruses, have been found in fruit bats. And it's thought that all other animals, non-human primates, antelopes, dogs, rodents, porcupines, pigs, and humans, are all considered intermediate or dead-end hosts. So you basically have the virus that circulates kind of within 3 different species of bats, as you see on the left. And then, incidentally, it causes secondary infections, both in intermediate hosts, in animals, and eventually in humans. This is basically what Ebola virus looks like. Some may think that it's actually a very pretty virus. Some may think it's really ugly. Perhaps those of you who have an artistic inclination, I actually think it's a little ugly, but that's kind of my opinion. It looks like a shepherd's crook, basically. And unlike other viruses, many of the viruses are very nice. They're symmetrical. They're pretty. This is a pretty ugly-looking virus that basically consists of, like other viruses, like a complex of different proteins. And what I have shown here, what's kind of amazing is that something as small as this Ebola virus, it's only about 9,000 bases in size. Something as small as this Ebola virus, which only codes for about 7 or 8 proteins, can actually produce such a severe disease. What I have here are basically, I show that these are the various proteins and how they're targeted by some of the experimental drugs that you've probably heard about that are being treated. ZMAP is a monoclonal antibody directed against the glycoprotein, shown there by those dots dotting the surface of the virus. That was used to treat actually the first 2 healthcare workers that came back from Africa. They were treated in Emory. Brincidofovir is a drug against the polymerase, which is the replication machinery or the replication protein for the virus. That was the drug that was used to treat actually the first patient who was admitted to Dallas, who was Liberian national. And then you have another virus called TKM Ebola. That virus was actually used to treat another doctor who came back to the United States with Ebola virus infection. This particular drug actually targets 3 different proteins. And actually, what's interesting is that this actually is a drug that was based upon the exact conception as the previous speaker. She talked about the idea of, can you generate basically something to either cut up the virus or inhibit it. These are actually, TKM Ebola actually are RNA molecules that simply bind to the virus and inactivate it, because this is an RNA virus. So the history of Ebola virus. This is what you see on the upper right. This is the Ebola River. It's named after Ebola River. It's basically found in Democratic Republic of the Congo. And the first epidemics actually occurred in the Congo. They were identified 2 species, Zaire and Sudan Ebola virus. And there have been, in addition, since then, that time, there have been additional 3 Ebola viruses that have been identified. So what is the disease that's caused by Ebola virus? It's called acute hemorrhagic fever. This is a disease that's usually caused by viruses. It occurs in tropical climates, usually transmitted to humans by arthropods, which are biting insects or rodents, or some unknown reservoir, such as bats. And it's characterized by high fever. You can see petechiae, which are the red spots shown there on the middle panel from bleeding into the skin, which can lead to internal bleeding, low blood pressure, and shock. Like, how many of you have actually read or heard about the Hod Zone by Richard Preston? Yeah. So, I mean, that's actually a somewhat sensationalized, as you might expect, account of Ebola virus infection. In fact, actually, the majority of Ebola virus infections don't have the bleeding. And the bleeding is a very, very late manifestation. When you start out with Ebola virus infection, really the symptoms at the onset of disease really resemble a really, really bad flu. So probably all of you have had kind of a flu-like illness before. This basically means fever, muscle aches, stomach pain, fatigue, and like intense fatigue is usually a manifestation. But you know, you could say that that's an early sign of Ebola virus infection, but it's also an early sign of influenza. It's exactly the same symptoms you would get with influenza. And this poses a diagnostic challenge, especially for, say, trying to screen, say, patients coming back from Africa by taking their temperature, because really it could be a number of diseases. It might not even be infectious that can cause fever. So how is it transmitted? Well, there's also been a lot of, I think, misconceptions about how exactly this virus is transmitted. All of our data really suggests that this is not airborne. This is not a virus that floats in the air, and basically, so if someone coughs into the air, it can potentially infect everyone in the room. This isn't that type of virus. There are viruses like that. Good examples are measles. There are other bacteria like tuberculosis, which are definitely airborne. This virus really requires contact with infected bodily fluids. And they include everything listed there. However, not every bodily fluid is the same. And in fact, when they looked at patients infected with Ebola virus infection, the highest titers are typically found in the blood, they're found in vomit, and they're found in stool. The virus can be found in saliva and can be found in sweat, but typically when it's found there, it's very, very low titer. Very low amount of virus is actually in these bodily secretions. And in fact, it's never been demonstrated that, for instance, sweat can actually transmit Ebola virus, although you can detect the virus in sweat. And that also basically makes it, what the other conclusion that you can get from this is that this is not really an easy virus to catch. I mean, it's a very scary virus to catch. And the fact that there's no treatment and there's no approved treatment or vaccine for the virus is certainly quite worrisome. But the big issue is that you can't really get it from casual contact. It doesn't appear to be a virus that's transmitted effectively by, say, an infected person touching a doorknob and then you touching the doorknob and getting infected that way. Okay, so this is basically this pamphlet that's spread by the CDC in terms of how you can get Ebola. You can really get it only by touching the body or bodily fluids or blood of a person who's sick of or who has died from Ebola. You can also get it through a needle stick, for instance. You can get it from touching infected fruit bats or primates, or actually in Africa, the common mode of transmission is actually ingesting fruit bats or primates. They typically, this is their source of protein, what they consider bushmeat. In the United States, you wouldn't have that problem, obviously. And I think what this slide actually highlights is the fact that the two groups most closely at risk of getting Ebola virus infection are very close household contacts of someone who has Ebola virus infection who are sick and is sick from it, or healthcare workers or healthcare providers, namely doctors and nurses. And I'll show you why I think that's the case. Okay? So one thing is that the information about how long does it actually survive outside the host. This is actually a labile virus. When I basically say 24 to 40 hours in liquid or dried material, that's usually a best case scenario. And typically, it's very rapidly inactivated by bleach, common household cleansers, any sort of alcohol-based hand rubs will immediately inactivate the virus, render it non-infectious. Sunlight inactivates the virus as well. And really, it's stable under conditions that you typically wouldn't find commonly, like in the dark at 4 degrees. So, I mean, if you take an infected Ebola virus secretion, put it in your refrigerator, I mean, you can keep it alive, but I don't know why you would want to do that, basically. But you could. You could also keep it indefinitely stable and infectious at negative 70 degrees, and that's how we use it to study the virus. And I think it's quite telling that in the 2 cases in the United States that were treated by, well, now it's actually 3, but in the cases treated at Emory Hospital, they actually did very careful environmental testing of all of the exposed surfaces by those patients. So basically where the patients had touched, including the toilet, bed rails, the doorknobs, kind of railings and surfaces, and they did not only did they not detect infectious virus, they didn't even detect the RNA, the nucleic acid from the virus, in the surfaces. However, when they screened the patient's vomit, stool, and blood, there were very high titers of the virus. Okay? All right, so how does the disease progress? Well, typically the incubation period can last up to 21 days. More commonly, it's 4 to 9 days. The reason you see the 21-day quarantine in the literature is because that's how long the incubation period can last. After you develop symptoms, it begins for days 1 to 3 as a kind of prodromal flu-like illness, which then can develop into vomiting, diarrhea, low blood pressure, and then finally the bleeding. And these patients actually die from massive blood and fluid loss. Every patients, they were losing 5 to 10 liters of fluid a day, and most of it was from diarrhea. So if you can imagine 10 liters of highly infectious diarrhea, and then you can basically consider, it doesn't make it can basically provide explanation, for instance, for how the 2 nurses got infected. I mean, there are huge numbers of, in very sick patients, there are huge numbers of highly infectious materials being made daily. I'm going to skip this. We've already gone over symptoms of Ebola, but another question, is there a cure? Unfortunately, there's no approved treatment and there's no approved vaccine. And so you might ask, well, why isn't there an approved treatment vaccine? I mean, we knew about this virus since 1976. And the unfortunate answer is that because this has been a virus that's only affected very few people, mostly in remote areas of Africa, it's not been a virus that's been judged of high priority in terms of research funding or even clinical funding. And it's very unfortunate that it's only now that we have such a huge global outbreak that's out of control that now there are efforts being made to provide funding to do the research to develop drugs and vaccines. And what had actually happened was that there are many vaccines and drugs that have actually been shown experimentally to be 100% protective and 100% effective in monkey models or animal models, but they've never gone to the human trial stage. That's usually a stage which usually requires, say, a $100 million investment. And unfortunately, there's no business model for this. Companies are just unwilling to pay up. It's understandable because they really have no good way of getting their money back to making that investment. This has been, and the problem with current experimental drugs, such as ZMapp, is that even though they're very promising, all of the data that we have is anecdotal. I mean, we've tried them on a few patients, they work, they appear to work, but we don't know whether or not it was because we gave them the drug that the patients got better, or because of other interventions like the supportive care, blood transfusions. There might have been, or maybe simply because they were lucky. There could be some reasons why. And this really highlights the importance of doing clinical trials, really to show whether or not these drugs are actually effective. So the mainstay of treatment turns out to be what we call intensive supportive care. And this is what's being done, for instance, for the current patients, the patient in New York, the patient from Dallas, that were eventually transported elsewhere. Really, because of the fluid losses, probably their #1 treatment is intravenous fluids. We have to constantly replete these patients with fluids, give them fluids. Because they also have such high fluid losses, they also have severe electrolyte imbalances in their potassium, sodium, calcium, and that can lead to cardiac arrhythmias or abnormal heart rhythms that can result in basically heart block and sudden death. So because of that, they also need electrolyte replacement. They may need blood transfusions, and they're given antibiotics not to treat the disease, but to prevent secondary bacterial infection. I'm just showing you a kind of a list of some of the drugs that have been tried. TKM, Ebola, ZMapp, and brincidofovir. There have been animal models that have shown efficacy for Tecmira, which is TKM, Ebola, or ZMapp. Brincidofovir actually was never previously tested in animal models for Ebola. It was actually, interesting enough, used for adenovirus infection, but it happened to have activity against Ebola, so they're considering the drug for that purpose. And then there's another drug that you may hear about. It hasn't been, to my knowledge, used yet in humans. It's called BioCryst, or BCX4430. This one actually targets the polymerase, which is actually common. It targets a region that's common among a wide variety of different RNA viruses. So in principle, in theory, it's possible that this drug is effective not only against Ebola, but against yellow fever, against Marburg, against MERS coronavirus, even against influenza. And it's being tested for that purpose. So to kind of summarize how to prevent transmission, asymptomatic patients who are Ebola infected do not transmit the disease. They need to have evidence of symptoms. They need to have a fever. They need to perhaps have some vomiting, because then their bodily fluids could be infectious. So the general principle of preventing infection in hospitals is to wear what we call personal protective equipment, or PPE, which is, as you might expect, are gowns, masks, gloves, etc. And new CDC recommendations are actually that your whole entire body needs to be covered. So the prior CDC recommendations were actually that you need to cover your neck, for instance, but now you actually have to wear a hood that entirely covers your neck. Many hospitals have actually gone, including ours, have actually gone to using essentially these whole body suits called Tyvek suits to kind of cover your whole body while you're taking care of an Ebola patient. Basic infection control measures are important, and then this other aspect of prompt and safe burial of dead, and we'll actually go into that because that's an interesting cultural moray in West Africa, is that they treat, part of their tradition is actually to properly take care of, properly dress, and to take care of the dead. And what by that means is that they have to undergo kind of specific burial rites or burial practices or ceremonies. And what that usually entails entails touching and kissing the dead body. And unfortunately, it turns out that a dead body can have about 10 billion copies of the virus per mil of fluid. Okay? So if you imagine, and it turns out that it's very likely that that was actually how the Ebola virus spread so rapidly through West Africa. And that's also why you're very unlikely to see, for instance, a same type of thing happening here, where the question is, could there be thousands of cases in the United States? And I think the answer is no, simply because this is how it sort of spread in Africa, and also the fact that the three countries really do not have adequate healthcare systems. But I think that the same thing is very, very unlikely to happen in the United States. Not to say that we're not going to see more cases, and in fact, we may see quite a few more cases, which will quickly overwhelm the capacity that we currently have. The 4 institutions that have been specially designated to treat infectious diseases like Ebola, which are institutions, basically Emory University, University of Nebraska, in Montana, and the NIH, they can actually collectively only manage 12 patients maximum. So if you think that once we get to 100 patients, we're going to be in trouble. Okay, so this gives you another idea. This is basically a published paper that was published by the CDC that gives you an idea of when are patients most infectious. And if you actually look here on the left, it actually turns out that at the very beginning of the illness, at day 0, you actually have a titer that's typically barely above the detection threshold. What you see here on the left is a logarithmic curve. But however, by day 3, the titers can actually rise to about 1 billion copies per mil. Okay? And so really, when you're most infectious is 3 to 7 days after the onset of symptoms. You're not really infectious at the time of diagnosis. And in fact, sensitive PCR, polymerase chain reaction tests at the onset of diagnosis can be even negative in someone that's infected with Ebola. The current recommendations are if you suspect someone in the hospital that have Ebola, you immediately isolate them, then you do PCR testing. You may have to repeat your test. In fact, the recommendations are you repeat your test 48 hours later before you can actually release somebody from quarantine that you have a strong suspicion may have Ebola. So let's talk a little bit about Ebola in Africa and how it quickly rose. This is kind of the current situation. This is as of this week. You can see here that the cases are really concentrated in 2 of the 3 affected countries, Sierra Leone and Liberia, although it did begin in Guinea. In fact, it did begin in the village of Guekedo in Guinea. And these are the cases in the United States and Europe. And pretty much summarizing, we have a total of I believe about 9 cases total that were diagnosed, that were either brought into the US or they're currently being treated. And we also have cases as well in Europe. So the big question is why did it happen? Why this outbreak? I mean, this thing, we've had 30 outbreaks of Ebola virus since it was discovered in 1976. Why is this worse? Well, one is called this phenomenon called deforestation. The big issue right now is that vast areas of the African jungle have now been deforested. In fact, in the region of West Africa, it's thought that 70% of the forest cover is no longer there. And what this does is it puts humans in direct contact with the animal reservoirs that carry the virus or presumably carry the virus. And it's in fact a study that was done in the Journal of Veterinary Research basically said the increase in Ebola outbreaks since 1994 is frequently associated with drastic changes in forest ecosystems in tropical Africa. That's thought to be a very important reason. Another is bushmeat consumption. We already said that this is their source of protein. This is how they basically kind of consume and get their protein in their diet to avoid malnutrition. And this is a way of life for them. So, and the problem is that many nasty diseases, unfortunately, are known to be originate from bats and rodents and kind of other things that they consume. And the other problem is that Ebola virus causes a hemorrhagic fever that's as infectious and as severe in non-human primates like monkeys and apes, which are consumed by humans, as they do in humans. So really by simply like skinning or butchering, say, a monkey, you may get exposed, and that actually is a relatively efficient means of transmission, basically through microabrasions in the skin. We've already talked about burial ceremonies, and then this was the other reason, is that prior Ebola virus outbreaks were in remote areas of Central Africa, where basically there's no transportation, there are no roads there. Many of these areas in Central Africa, you can only get in either by motorcycle or by plane. And in fact, we're actually studying samples now from another outbreak in Central Africa that's occurring right now in the DRC, Democratic Republic of Congo. And those samples actually, I have to sort of pay for the plane flight to actually have the samples transported to the capital Kinshasa and then transported to my lab. And it's actually like $5,000, a ridiculous amount of money, just to get the samples basically to you. But however, in West Africa though, if you look here, there's a very nice road which, in hindsight, unfortunately facilitated the spread of the virus. It began in Guécado, it then went all the way west to Conakry, which is the capital of Guinea, and then from there it spread into Sierra Leone and Liberia. There's an extensive network of roads. So whereas they don't have a very good healthcare system, they actually have a very good transportation system, unfortunately. Okay, and the other issue is this issue of inadequate medical personnel. So what you're seeing here is this is actually the hospital. Okay, this is the Ebola ward. Okay, to give you some idea of the kind of conditions that you're seeing here, there's one doctor per 100,000 population. That was before the Ebola outbreak. Okay, the other thing is that they've estimated lost about 30% of their doctors to Ebola, unfortunately, and they have died at these hospitals. The Kenema Hospital, which is an example of one of the hospitals there. So you see here that we really don't have enough support, and really it's quite significant, basically, kind of this lack of resources. There's also been this question of, like, can Ebola mutate? This is an RNA virus. We know it mutates. RNA viruses do mutate. They change. Their nucleic acid sequences can change. There's also been kind of the worry is, can this virus mutate to become airborne? Because if you think about it, if you have a transmissible airborne virus that has a virulence of Ebola, I mean, that's the hallmarks of a pandemic. I mean, that's kind of the nightmare scenario that everyone's worried about. What may be a little more comforting, though, is the fact that viruses can mutate. And indeed, probably the most common kind of mechanism by which they mutate and end up affecting kind of the course of the disease is one is that they can adapt to the new host. So for instance, the idea would be that we're worried that Ebola may mutate so it becomes, ironically, less virulent. Because the problem is if it becomes less virulent, then patients may survive long enough, or they may survive completely, they may survive long enough to actually transmit and pass the infection to others. So that would be what we call viral adaptation. That can happen. Certainly you can have viral evasion, where the virus mutates so that the current tests that we're using to detect the virus no longer work. And then viruses are also known to mutate to become resistant. So a good example is HIV, for instance, where the virus can mutate, and then so that drugs and vaccines are no longer effective. However, what's comforting is that I think that the probability that it actually mutates to become airborne is close to zero. And the reason is because in the entire history of RNA viruses, no RNA virus has ever mutated to change its mode of transmission. Now, in theory, it's possible. It certainly is possible. And in fact, there are scientists doing what we call dual-use research that are studying, for instance, changes that you need to make influenza or bird flu, infectious in humans. But whether or not I can't say there's zero probability, but I can say it's probably very, very low. And I think that the worry about it is probably a little overblown. The other thing that was described is, this is a virus that has a huge number of mutations. In fact, they're quoting as 300 plus mutations. That is true. It actually has 395 mutations in the genome. However, when they're talking about that, they're actually talking about the differences between the 1976 strain and this current strain that's circulating. When you actually look at the 2014 West African strain and you look at the diversity of mutations within that particular strain, you're only talking about 15 coding mutations. So largely, this is the same virus. It's not a virus that's mutating like mad you're generating normal viruses. The same virus that caused the infection in the New York patient is the same as the virus that caused the original case in Guinea in last year, December of last year. So I think that a coordinated global effort is urgently needed. This is really a consortium of academia, industry, and government. And what we're doing is we're working on better ways to diagnose Ebola. Currently the gold standard is what we call polymerase chain reaction, or PCR, where we're using primers and probes to look for one specific region of the virus. There are also some attempts to use what we call rapid point-of-care testing. These are called lateral flow immunoassays, where you can just take a drop of blood or urine, and the idea is that with a pinprick you can diagnose whether or not a patient was infected with Ebola within 10 to 15 minutes. But we're really interested in also applying new technology to do this. And one big problem, as I mentioned before, with diagnosis is now in West Africa, now they're starting to see what we call Lassa season. Okay? Lassa fever is a virus, or Lassa virus is a virus that causes a hemorrhagic fever, very similar to Ebola. It's not as bad. Only about 15% of people infected with it die, so probably still wouldn't want to get it. But it's not as bad as Ebola. But one big problem is you don't know whether or not now someone has Lassa fever. This is in West Africa. Whether someone has Ebola, whether someone has malaria, whether someone has influenza, whether someone has TB, whether someone has infectious salmonella or typhoid fever or dengue virus. So the big issue is that there are all these diseases that cause the exact same clinical presentation. And unfortunately, the big problem right now is in areas like Kenema, which is Sierra Leone, you have basically patients, like Ebola patients are now sitting next to Lassa patients, or at least in kind of their waiting room right now. And I actually have pictures of that, unfortunately, which is, and so you imagine, we need a way to do what we call multiplex diagnostic testing. To have a single test that can detect everything or every potential agent. So we're using a method called deep sequencing. It's called next generation sequencing. The general idea is a brute force technique. All we're doing is taking clinical samples, generating millions of sequence reads from it, and we're basing the approach is based on the fact that all viruses, bacteria, parasites, and fungi are made of DNA. So in principle, why can't you just look at the DNA, be able to diagnose a patient? We've actually used that recently. It was a case that was reported in the New England Journal of Medicine just last month, where we used it to diagnose a case of leptospirosis in a 14-year-old child who actually returned from Puerto Rico. He had encephalitis that was unknown, was admitted to the same hospital 3 times over 4 months. They basically spent like $300,000 on his hospital care. And we basically did this test, and we were able to find the cause of it, basically. It turned out when he was critically ill, and we were able to use it to potentially save him. But the general idea is, can we use this technique to be able to identify viral infections? And so this gives you an idea. These are actually the first 8 cases in a different outbreak in Central Africa that's ongoing. This outbreak actually began in August of 2014, so this is very recent data. These are the first 8 cases. And if you look here on the 3rd column, you'll see by real-time PCR, that's the gold standard test. We only detected the virus in 4 of 8 cases. If you use conventional PCR, which is what we had before, it's 3 of 8. But we're able to use what we call deep sequencing, and the advantage of deep sequencing is we're not looking just for Ebola. We're actually looking for Ebola, Lassa, or anything else. And we were able to diagnose it actually in 6 of those 8 patients. And you can see here, what's nice about this particular test is because we get sequence information, the software that was developed in my lab to do this can immediately not only tell you that this is Ebola, it can give you the closest genome in the database. It can tell you what strain or species it is. You can actually get sequences or do what we call whole genome assembly, sequence the entire genome. You can look through the genome. It'll let you know whether or not there are mutations in the genome, whether resistance factors, other things that will give you some guidance as to what exactly is causing the disease and how best to treat it. In addition, there's a big problem right now with diagnostics in that this is basically taking words from Dr. Frieden from the CDC, that basically there's no way to detect the virus prior to symptoms. And that's been a big problem, because if you imagine patients coming back, or potential patients coming back from Africa, screening for, say, just temperature checks and history, I mean, while it's probably a good thing, it's probably not sufficient. Because, as we just seen from this week, there are cases that will kind of pass, patients that are asymptomatic. There's no good way right now to rapidly diagnose them with Ebola. So what we're developing is, we're actually hoping to find what we call a gene expression signature. The idea is that patients who are infected with Ebola, even though you can't detect the virus yet, you may be able to detect the fact that they've been infected by looking at the pattern of host response. Now, this has actually been shown to be effective in influenza, for instance. A group at Duke University, they were actually able to look at the pattern of gene expression, be able to diagnose someone with influenza without looking for the influenza. And it turned out that the signature for influenza was actually very specific for influenza and didn't overlap with other febrile illnesses. Now, we've actually been also doing some work with acute Lyme disease, and where we can find that there is also a gene expression signature for diagnosing acute Lyme disease. We have a paper that we've submitted just recently which suggests that you can use a panel of just 60 genes, can tell you whether or not you have acute Lyme disease and distinguish it from some other viral or bacterial infection. So this is very preliminary data, and actually we just generated this week, so take it with a grain of salt. But the general idea is we looked at the same 8 patients, and basically the 6 patients that we believe actually were PCR positive and really had Ebola infection, you can see basically clustered together in those red circles over there. And it appears that the clustering is independent between normal patients from plasma and whole blood, as well as between other causes of febrile illness in Africa, including Lassa and malaria. And we're currently actually doing this now in collaboration with the Department of Defense to develop a rapid assay to look at the host response, but also look for the virus at the same time, and to be able to diagnose or better diagnose patients that may have been infected with Ebola. So I just want to thank some of the people in my lab. Sammy Nakash and Jerome Bouquet are 2 postdocs in my lab who are doing all of the Ebola virus work. I have a number of collaborators both in Africa and in France that are working with me on Ebola, and we're being funded by the NIH as well as by an Avid Viral Discovery Award. So I'd be happy to take your questions, and thank you for listening.